I'm Daniel Levine, and this is the Bio Report. Though cancer vaccines have been an area of great promise, in practice they face several challenges because of the heterogeneity of tumors, the ability of the tumor microenvironment to suppress the immune system, and the challenges of producing a strong and sustained T-cell response. OSE Immunotherapeutics off-the-shelf cancer vaccine Tedopi has shown promising results in a phase 3 study in patients with non-small cell lung cancer, and the company is now conducting a confirmatory phase 3 study. We spoke to Nicolas Poirier, CEO of OSE Immunotherapeutics, about the company's off-the-shelf cancer vaccine, non-small cell lung cancer, and how it's leveraging its immune system expertise through partnerships with leading pharmaceutical companies. Nicolas, thanks for joining us.
Hi, Daniel. Thank you.
We're going to talk about cancer vaccines, OSE Immunotherapeutics, and the company's broader efforts to address immunoinflammation. Before we talk about any specific programs, though, I wanted to begin with OSE itself, which is not only pursuing immuno-oncology therapies, but treatments for immunoinflammation as well. In one case, you're seeking to stimulate the immune system, and in another, you're trying to quiet it. Is there any interplay between these efforts? Do you learn anything from one that informs the other?
Oh, yes, we learn a lot from one field to the other. So I think immunology is our playground. We love to explore what's wrong in inflammations, autoimmune disease, why our immune system is attacking our own tissue. And on the opposites, we learn a lot how we can eradicate or redirect our immune response against the cancer cells, against the tumors, how these cells are capable to hijack or pirate our immune system. And again, vice versa, by studying what's wrong, why the immune response is not working very efficiently in autoimmune disease, how the tumors inhibit the immune response, we can understand how we can manipulate, control immune response against our tissue in autoimmune disease. So it's really, we are really learning from one field to the others. Yeah.
OSE does have multiple platform technologies, and it's pursuing a number of first-in-class assets and novel approaches to modulating the immune system. I do want to focus on your cancer vaccine, but let's touch on some of these platforms through a program for each. Can you start with the BICI platform and your experimental therapy, OSE 279?
Yeah, sure. So OSE 279 is our own proprietary anti-PD-1 antibody, similar to the pembrolizumab or nivolumab, the so-called PD-1 inhibitors that revolutionized immuno-oncology. And we used this own anti-PD-1 as a backbone platform to deliver new biology for the patients with unmet needs, meaning the one that do not respond currently to the immunotherapy. So we can graft or fuse, if you want, other immunotherapeutic modalities such as cytokines, such as other immunostimulatory molecules to our anti-PD-1. And by doing that, we are not just developing a bispecific or bifunctional approach. We are really looking for synergistic activity between the two modalities, meaning we can redirect the cytokine or the second modality to the PD-1 expressing lymphocytes, which in cancer patients are the tumor-specific T cells. So the objective at the end is to really target the right part of the immune system, the right T cells at the right place to have more efficient, more targeted immunotherapies with the objective to increase efficacy but not increase toxicity, because this is a drawback today of current combination strategies.
OSE also has a myeloid checkpoint platform that it used for OSE 172, which is a signal regulatory protein or SERPA alpha targeting antibody. Can you explain what that is?
Yeah, for sure. So OSE-172 is an anti-SIRP-alpha monoclonal antibody. And SIRP-alpha, if you want, is similar to PD-1. So PD-1 is a major brake of T lymphocytes. This is the part of the immune system that is really adaptive. There is a second part of the immune system that has not been covered by the immunotherapeutics right now, which is the innate immune system. So the innate immune system is really the basic historical immune cells capable to eat the infected cells, or the cancer cells. And the major brake on these cells is named SIRP-alpha. So we have the capacity, like PD-1 inhibitors, that release a brake on the T lymphocytes. With the SIRP-alpha antibody, we can release a brake on the myeloid cells. And the beauty is that we can combine these two approaches. And in that situations, we are capable to release the two major brake on the two big parts of the immune system, again, to look for synergistic activity for the benefit of patients.
Finally, OSE has OSE 230, which emerged from your pro-resolutive monoclonal antibody platform. Can you talk about 230 and this platform?
Yeah, it's a very different platforms. Now we're moving to the inflammation side, the other side of the mirror. So what we learned this last decade that the majority of the patients, unfortunately, are not well managed by the current therapeutic approach, and mostly because they are all targeting the same type of the immune system. We understood how we can induce trigger resolution of inflammations by targeting other immune cells like the neutrophils or the macrophages again. And so OSE-230 is the first antibody from our pro-resolutive monoclonal antibody platform that target KMR23. And KMR23, the target of this program, is a really fascinating target that is expressed only in the tissue by neutrophils. So we have a quite unique opportunity with this program that we just recently licensed to AbbVie to target the neutrophils, the chronic neutrophilic infiltrates that are pathogenic in tissue, in inflamed tissue, while sparing the neutrophils in periphery that are really precious, really important for our own safety to fight against bacteria.
OSE's lead experimental candidate is a therapeutic cancer vaccine. Immunotherapies have had great success in cancer, but therapeutic cancer vaccines have not yet realized their potential. It's about 15 years since the approval of Dendrion's Provenge. What's limited the success of cancer vaccines to date, and what makes them challenging to develop?
Yeah, I think it's difficult questions. I think today we know better the patients, and thanks to the revolutions of the anti-PD-1s and immunotherapy in oncology, we know better the patients. We know better how the immune system works, not globally, but precisely in the tumor microenvironment, which is a very particular, let's say, ecosystem. We have learned a lot about that this last decades or 15 years. And in parallel, there was a dramatic improvement in the molecular engineering capacity. So now we know better what antigens are the right targets to improve or to eradicate the immune system. And we know how to make these antigens better to really stimulate strongly and on long term the immune system. So I think that's the two key most important elements that will guarantee the success of the therapeutic cancer vaccines that are currently under development by us and by others in other indications. Yeah.
The company is preparing to begin a pivotal phase 3 trial of its Tedopi cancer vaccine as a treatment for non-small cell lung cancer. What is non-small cell lung cancer, and how big an unmet need does this represent?
Oh, it's huge. Yeah, we have already started the phase 3 a few months ago now in Europe and US. Non-small represents 85% of the lung cancer bracket. So it's a really huge. It's the most prevalent lung cancer type, and lung cancer is the second cancer type in terms of incidence after breast cancer. But unfortunately, it's the first cancer in terms of mortality. It represents more than 2 new millions case per year in the developed countries, let's say Europe and US. So it's really, really huge. And even if the immunotherapy, the so-called PD-1 inhibitors, have been recently approved in lung cancer as well, the majority of the patients, more than 60-70% of the patients, do not respond and have no therapeutic options. So the medical needs are still really huge in lung cancer as well.
What treatment options exist today, and what's the prognosis for someone diagnosed with the condition?
Yeah, it really depends on the stage of the disease, let's say for lung cancers. When you are diagnosed, of course, if it's at very early stage, non-metastatic disease, that's very different from the non-small cell lung cancer with metastasis that we are treating. So the first line now for metastatic disease is a combination of chemotherapy plus immunotherapy, so the so-called PD-1 inhibitors. And then in second line, so when the patients do not respond or unfortunately lose a response, there is nothing approved after. There is nothing that significantly demonstrated efficacy in pivotal registration study, and that's why it's so important for us to conduct this study in these indications.
You touched on some of the challenges cancer vaccines have faced, particularly around the tumor microenvironment and its ability to suppress an immune response. How does Tedopi address that?
Yeah, so Tedopi is currently exploring different cancer types that are quite representative of different tumor microenvironments. So currently we're running this pivotal phase 3 in lung cancer that is considered a, let's say, hot tumor microenvironment like in melanoma. On the opposite, we're also conducting phase 2 in pancreatic cancer that is the extreme, really cold tumor microenvironment with more difficulty for the immune system. In the between, we have the ovarian cancer that we're also exploring in phase 2. So right now, we are conducting several studies in different type of tumor microenvironments to really explore where the vaccine is efficient. We know it is efficient in lung cancer. Now we need to demonstrate in the formal pivotal registration study. And then we will know this year and next year with the ongoing phase 2 whether we can also extend the potential to different type of cancers and different tumor microenvironments.
This is an off-the-shelf vaccine. What does it consist of, and how does it work?
Yeah, off-the-shelf means that the vaccine is ready to use. So that's really important in the field of cancer vaccines. There is right now 2 big approaches that are complementary. One is personalized, the other one is off-the-shelf, ready to use. We are targeting, for example, in non-small cell lung cancer, metastatic patients, as I said, and metastatic patients that lose or that do not respond anymore to the first line of treatment. So the patients, unfortunately, are under progressions, meaning their tumor is growing every day. We have no time to waste, no time to lose to manufacture a personalized vaccine for several weeks. In the better case, it's 3-4 weeks, sometimes could be more. So we need, and the physicians want the treatments right now, the day after. So that's a big advantage of being off the shelf, ready to use for this particular type of patients. In other situations, like in the personalized RNA vaccines, this is applied after surgery. And the idea is not to attack directly the tumors or to induce reductions of the tumor size. The idea is to prevent relapse, regrowth after surgery. So this is 2 complementary approaches at 2 different stages of the disease.
How do you determine the epitopes, the portion of antigens in the tumor that are needed to make an effective vaccine?
Yeah, this is something that we have done several years ago, to be honest, in the labs, with the idea of if we identified one super antigens on a tumor, the risk is that the tumor can mutate, generate variants like virus, and then escape to the treatments. We know that very well. So the idea was not to identify only one tumor antigen, but to identify several shared tumor antigens. Shared means we need to make sure these antigens are really commonly expressed by tumors across patients, because of course each patient has a different tumors, and we need to make sure that we don't want to select the patients for expression of the antigens, and it's not personalized. So we need to make sure that these antigens are expressed by 70-80% of the patients. And we also wanted from the beginning to design a vaccine that could be applicable for lung cancer, but also ovarian cancer, pancreatic cancer. So we looked for not only shared tumor antigens across patients, but shared tumor antigens across different cancer type, different indications. And so we identified 5 of these tumor antigens. And within these tumor antigens, the point was to identify the most immunogenic parts, what we call the epitopes of these tumor antigens, to stimulate the immune system. This has been explored quite a lot, decades or 2 decades ago, and it mostly failed because these tumor antigens are from our own cells. So it's considered by the immune system as a self part of our body, while the immune system is trained to recognize non-self, pathogenic. So in our platforms, we engineered these epitopes, we modified them, we create what we call neoepitopes to make them more different, more immunogenic, and these epitopes are stronger to stimulate, eradicate the immune system to recognize cancer cells and generate T cells that are armed to kill the tumor cells when they have recognized these cells.
The vaccine isn't expected to work on everyone. It depends in part on the individual's immune system. In the case of Tedopi, patients need to be HLA-A2 positive. Can you explain what that means and whether you expect to use a companion diagnostic to identify patients appropriate for the therapy?
Yes, indeed. So these epitopes recognized by T cells are not recognized alone. They need to be present to the immune system. And the molecule that present these epitopes to the immune system are named HLA. And there is a lot of polymorphisms in this HLA system, meaning that you and me, we don't have exactly the same HLA. And so if we graft my kidney to you, for example, you will reject this kidney because we don't have the same HLA system. So this was a big challenge, and this is still a big challenge if we want to develop a vaccine off the shelf, ready to use for everybody. Hopefully, there is one HLA molecule named HLA-A2 that is commonly expressed by nearly 45% of the populations in Europe, in US, a little bit more in China. So we have engineered, as I said previously, the epitopes to be stronger for binding to these HLA-A2 molecules. So it means that if the patients express this allele, this HLA-A2, it is a biological responder to the vaccines. So now, in our clinical developments, after discussion with the FDA and EMA, we have developed a regulatory strategy with a companion diagnostic to select only HLA-A2 patients. But it's really easy, just a blood sample and a sequencing of our bloods. We are HLA-A2 genetically positive or negative. If you are positive, you are eligible to receive the therapy because we know that you will be a biological responder, meaning your immune system will be educated by this vaccine. And then we can enrich in responders to explore the efficacy in cancer.
What's known about Tedopi from the studies that have been done to date?
So we ran previously, of course, phase 2, which was positive in lung cancer patients, and then a first phase 3 in third line in non-small cell lung cancer that we published last year. This study was really interesting, really informative, because we learned that there is, in the patients that are refractory right now to the immunotherapy, there is nearly half of the patients that initially respond or have benefits, but secondary lose the response. This is what we call secondary or acquired resistance. And what we have demonstrated in our previous phase 3 that these patients, if they receive the vaccine alone in monotherapy, they survive longer than if they receive chemotherapy. So it means the vaccine alone is better than chemotherapy. At 1 year, the survival rate was 44% for patients receiving the vaccine versus 27% only for chemotherapy. So that's the first thing that we learned. The second one but this was more anticipated, the patients feel better. They have 3-fold less toxicity when they receive the vaccine compared to chemotherapy. So of course the toxicity profile has nothing to compare with the chemotherapy. And this translates in better quality of life for the patients. So based on patient-reported outcomes, the patients score themselves on different scales clearly better. So the patients survive longer and better when they have developed this acquired resistance to immunotherapy. And now, after discussion with the FDA and EMA, we validated the regulatory path to move for registration with this new confirmatory pivotal trial, these phase 3 that we have initiated now in second line non-small cell lung cancer in US, Europe, UK, and Canada.
You're exploring this both as a monotherapy and a combination therapy. What would you expect it to work well with in combination?
Yeah, the point with combinations, as always, is to see whether we can increase the efficacy. The hope or the dream is, of course, to cure cancer for the majority of patients. And we know that some patients will respond to the vaccine as a monotherapy. And that's really important because monotherapy, you have no doubt where comes the efficacy, the activity. When you are in combinations from the beginning, you can have great results, but you never know which part of the combinations drive the efficacy. So we know our vaccines works in monotherapy, works better than chemotherapy. So we are sure about that. Now the question is, could we increase their efficacy for the benefit to more patients? And that's why right now we're exploring in 3 independent ongoing phase 2 combination of this vaccine with the so-called anti-PD-1. So we are combining with nivolumab or pembrolizumab in non-small cell lung cancer or ovarian cancer. And in pancreatic cancers, that's really different. We are running a phase 2, but in these indications, we know the anti-PD-1 are not working. Chemotherapy is really important to modify the tumor microenvironments, improve the response. So we combine the vaccine with chemotherapy, and we're expecting the result of this phase 2 now this year.
OSE has collaborations with a number of major pharmaceutical companies. I'm wondering if you talk a little about how you think about the role of these partnerships and what beyond revenue you look for in a partner.
Yeah, they are central in our strategy, at least in our business model. We have 3 ongoing large pharmaceutical partnerships with AbbVie, Boehringer Ingelheim, or Veloxis. They're really important because on one side, it validates the technology, the science. Then these companies will or are in charge of the developments and in terms of cost. So it's they are in charge of the cost and also of the risk of these developments. So for us, it's only pure future potential revenue. We are paid in these contracts through an upfront plus milestone based on success and royalties. That represents more than 80% these last decades of our revenue. So 80% of non-dilutive cash that we re-inject in our own proprietary programs to develop more value and develop our proprietary pipelines.
The AbbVie agreement provided $48 million in upfront payments and an additional $665 million in potential milestones. This is for OS 230, the pro-resolutive mAb we talked about a little earlier. What is OSE responsible for, and is there some decision point that AbbVie would have to make and take responsibility for it?
Yeah, so right now, we license this program yearly in 2024 to AbbVie. This is a preclinical program, or let's say pre-IND program. So meaning that we're in charge right now of manufacturing the GMP batch, so the clinical batch. We're in charge of the transitions to the clinic, all these studies, finalizing toxicology and translational studies and so on. And then when the molecule will enter in the clinic, let's say the IND, AbbVie will be in charge of all the further clinical developments, and we will be paid through this system of milestone at each stage they will success.
In May, Boehringer Ingelheim expanded its collaboration with OSE with the addition of 2 new first-in-class treatments for cancer and cardiorenal metabolic diseases. This is on top of the existing work you were doing with the company on anti-SERP alpha compounds. Can you talk about the significance of this and what it means for OSE?
Yeah, this is a significant partnership that we have signed in 2018 with Boehringer for more than potentially 1 billion of milestone and of course royalties. So that's really significant for us. We have 3 partnerships, a potential of 2 billions, and Boehringer is already half of that. Initially, we licensed this program for similarly to AbbVie at the preclinical stage. We were in charge similarly of the transition to the clinic, but also the first clinical investigations because from the beginning we understood we will be more agile, rapid to explore the efficacy and safety in clinic. Now Boehringer is in charge of the other step in terms of clinical developments in oncology. And more recently, last year, we renegotiated our contracts because we found that there is great potential for the same therapy, the same anti-SERP alpha antibody, not only in oncology, but in this cardiorenal metabolic space. And mostly they start now, this is official, their phase 2 study, so efficacy study in MASH, hepatic metabolic disease. So that's really interesting. Coming back to your first question at the beginning, we can learn from immuno-oncology to immunoinflammations, but now we know that we can learn a lot about these 2 therapeutic space for other therapeutic space like metabolic disease, where the immune system has some limitations in controlling the metabolic disorders, and by manipulating the immune system, we can really provide benefits for the patient. So they have started their phase 2 in MASH indications, and they pursue the developments in oncology. So it's only benefit for us. More therapeutic indications, potentially more patients, and in, as you know, metabolic disease, it's a huge, very huge market size.
How do you balance your internal pipeline with advancing work for partners?
Yeah, this is an excellent question. We're lucky, let's say, we are rich in terms of capacity to generate really interesting innovations. On the opposite, we are scientifically rich, but it needs a lot of money to develop all of these molecular innovations. So the way we are deciding is clearly depending on the opportunity. We think that when we are targeting a really large indications that will require huge investments, if we can validate the science, if we can de-risk the pipelines with a partner, that's really good for our company and shareholders. And on the opposites, sometimes when we have a more targeted market approach with a companion diagnostic like the cancer vaccines, we can go further until registrations and finally at the end find a partner for the commercializations. But we are completely in charge of all the development activities.
And are you expected to build a commercial team, or will you seek partnering for marketing all of your products?
We really think at OSE that we are strong to innovate. We are strong to explore how we can eradicate our immune system. We are agile and probably faster than the large companies to demonstrate safety and efficacy in clinical developments. But where the large companies are really strong is for commercialization, distribution, and we think that it's a benefit for everybody, our company, the patients, if we can find the right and appropriate commercial partners to accelerate and the access of the innovations to the patients after approval.
Nicolas Poirier, CEO of OSE Immunotherapeutics. Nicolas, thank you so much for your time today.
Thank you, Daniel.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.